Okay, so a good afternoon and a very warm welcome to this session in collaboration with TTP. So One Nucleus is delighted to be collaborating today with TTP. The value that the collaborations bring to our network is really important, because this is the way in which we bring expertise from other groups and companies to our membership, which enables our members to grow their own businesses. And even because this webinar is open to all, it's inclusive, so members and non-members as well. So before I go into introducing the speakers who will be joining us today, just a few words on logistics. So we are a fairly small group, as you'll see, so I'm not too worried about interfering sound or anything, but just to remind you, whilst the presentation's going on, please do remain muted. And as you will have heard, we are recording the session. So the session content will be disseminated post-session. So if you wish to catch up and then revisit some of the material, you can do so. So I just wanted to do a brief intro for the speakers that we have joining us today. So firstly, we have Dr. Dan Strange. So Dan is a biomedical engineer at TTP, focused on the development of automation and tools and technologies for cell and gene therapy manufacturing. He has worked in both the medical device and life science sectors, taking a leading role in projects ranging from the development of life-saving connected drug delivery devices to the development of GMP-ready bioprocessing equipment. He was also involved in the development of Covent, which was TTP's rapidly manufactured ventilator for COVID-19. And so Dan is also joined by Dr. James Lazarus. James is a consultant in TTP's Life Science Business Unit, where he mainly focuses on in vitro diagnostic product development. His technical background is in optical physics and laser system design, and he has led a range of complex multidisciplinary development programs. James's responsibilities and expertise include technical development, quality management, project management, and he's currently leading the development of a revolutionary digital pathology product. So we're in really good hands on the session this afternoon. I mean, we're a small group, so if anybody did want to take the chance to unmute and before we begin, just explain or just maybe introduce themselves and say what they hope to gain from the session, then we would welcome that. You don't have to, but there's an opportunity now if you wish to introduce yourself and just say maybe why you joined and what you hope to get from the presentation. So I'll just go silent for a minute to see if anybody wants to do that. There's no pressure, of course. Alternatively, please feel free to use the chat box to introduce yourself. So the chat box is enabled, so you can drop your questions and comments into that, and I'll keep an eye out and I can make sure that they get through to Dan and James post presentation. So with that, I'm just going to mute myself now. I'm going to hand the floor to Dan and James to take it away. So thank you very much.
Great, thank you very much. Just to give you a bit of overview about what we're planning to cover today. First, we thought we'd just give you the very 2-second intro of TTP, who we are, so you know where we're coming from and the backgrounds that we bring. And then really diving into, firstly, the different regulations that are out there. What are we going to be talking about today? In vitro diagnostics, GMP bioprocessing, things like that. Talking on some of the reflections for why regulation is there, before really diving in, and James will take us through a project plan of what it really takes to do a regulated hardware development and some of the insights and challenges and opportunities we've seen along the way, trying to ground that with some case studies to make it a little bit more interesting. James will talk through some in the diagnostic space, I'll then give some reflections on how that differs for GMP equipment, what's important there, and particularly maybe what's important as you start to think about cell and gene therapies and the differences there, before rounding off with talking a little bit actually about the rapidly manufactured ventilator, just in can you do these types of developments, regulated developments more quickly, with some top tips. After that, hopefully we can open up the floor a little bit, get some of your questions and get a good open discussion going of where there are, where you'd like to hear more. So just to give you the very quick background as to TTP, where we're 240 scientists and engineers based near Cambridge in the UK. We do technology and product development for our clients on a consultancy basis. And we do about 200 different development projects a year across a wide range of different fields. But a lot of that is really at that intersection with the life sciences, healthcare, and that inevitably also brings in regulation and what's needed. So we've worked on things ranging from implanted medical devices to cell sorting, to life-saving drug delivery, DNA sequencing, a lot of diagnostics work recently, as you can imagine. But actually, although most of what we'll be talking about today is hardware, that goes all the way as well to thinking about the interfaces with software, and we've done standalone software developments as well that have been software as a medical device. So hopefully that gives you a bit of a feel of where we're coming from. But as I say, I'll keep it relatively brief on the TTP so that we can more talk about doing regulated hardware development. Okay,
what do we mean by the regulatory environment? So when we think of the regulatory environment, we think of regulations and directives and also standards. And really the sort of difference between the two is that the conformity with the regulations is very much a legal requirement for the product manufacturer to enable them to sell their products in a given market. And the regulations that you must conform to depend on where you want to market your product and the type of product that it is. Whereas compliance with standards is a convenient way of demonstrating that you conform to the regulations. So in the EU, for example, in the field of medical devices, we have the new medical device regulation and the in vitro diagnostic regulation, and lots of other regulations that cover different products, such as machinery directive and low voltage directive, and also, automotive, lifting equipment, all sorts of different things. In the US, they have the Code of Federal Regulations, and medical devices and products are covered by Title 21 of the Code of Federal Regulations. So you'll see 21 CFR, and then the following code kind of defines what kind of product that is. So the regulations are the legal requirements the manufacturer must fulfill. Whereas the standards, as I say, are a kind of a convenient way of demonstrating compliance with those regulations. And harmonized standards are particularly helpful because if you comply with a standard that's harmonized to a particular regulation, then you essentially comply with all of the requirements of that regulation. So, for example, ISO 13485 is a quality management system standard, and that's harmonized to the in vitro diagnostic regulation. So if you can show that you comply with that standard, then you meet all of the requirements of the quality management requirements of the IVD regulation. And so in our business, that's kind of where we focus. We focus on developing the hardware or the instrument or the reagent or whatever it is that's in the product to comply with the standards that are identified by our clients. And our clients typically then go on to be the product manufacturer, and it's their obligation to comply with the regulations. So developing hardware in a regulatory environment for us typically involves a lot of focus around those standards. And so I've already mentioned 13485, which is the well-known quality management standard, but there are lots of others that in the medical space and the diagnostic space we need to comply with. So 14971 is a management one, for example, and then lots of other technical ones as well, to do with usability engineering or software development or electrical safety. And so that's typically where we focus, is developing the product in compliance with those standards. Okay. Did you take over here, or?
Yeah. So I mean, I think, you know, there's the different, whether it's a medical device, whether it's a in vitro diagnostic, or whether it's GMP equipment. I guess one of the questions is, well, why is that regulation there in the first place? What's it doing? Is it just a lot of paperwork that needs to be jumped through? Fundamentally, it really comes down to risk. I think we're all familiar with the image on the right, you know, computers regularly fail. A lot of electronic consumer devices that we have regularly just break. That's accepted, and as long as it's not happening too often, no one gets that too grumbly. Although sometimes people do get a little bit grumbly about when the blue screen of death actually appears. It's a very different story if actually the hardware, the device that you're making, when it stops functioning, can harm a patient, can harm a individual in the end of it. It's no longer acceptable for those risks to materialize. Unfortunately, actually, a lot of the regulation that's in place has really come out through a history of various public health incidents. So I think everyone's familiar with thalidomide back in the 60s, which then went and had a very significant effect on the way drugs are developed following that. Around a similar time in the 70s, there was an incident, which I'll talk through a bit later, called the Devonport disaster, where procedures and protocols around use of an autoclave led to, and the fact that those were not consistently followed and documented, and things were done verbally, led to the deaths of 5 people because infusion bags were not actually properly sterilized. That then had a big impact on GMP regulations. Those examples are, you know, decades in the past, but actually there are more recent examples which are driving behavior today. The PIP scandal, or the PIP scandal with breast implants 10 years ago has fed into the recent medical devices regulation in the EU. And if you're familiar with Theranos in the US, that's raised some questions in the FDA's mind about how lab-based diagnostics are regulated, and whether there's too much of a loophole there. So regulation really is about ensuring safety at the end. It's about ensuring reliability, consistency, so that when you're making something that is at the end having an impact on someone's life, it works the same way every time, and it's not going to suddenly stop or work in a different way, which then causes a harmful effect. And then lastly, risk benefit, because you can never actually eliminate all of the risks. So making sure that when you are working with these types of devices, the benefits actually outweigh the risks. Practically for us as developers, and we'll talk in a moment about what that really means, that's through quality and risk management.
Sorry, I advanced the slide too early there. I've got a whole load of really off-putting windows that have popped up on my screen, and I'm trying to close it. It won't let me. Okay, so what's different about regulation and the impact that it has on medical products compared with non-medical? So as Dan mentioned, when we talk about safety in medical products, we're not just thinking about safety to the user, we also have to consider that it functions correctly. So if you take the case of an in vitro diagnostic system, for example, not only does that need to be safe to use by the user, it also needs to return the correct information, because the clinician at some point is going to probably make some kind of clinical decision based on the output, and that output, they need to have high confidence that that's correct. Otherwise, they could be placing the patient at harm. So there's a key difference. And that is very much baked into the whole development process. And so what we need to do when we develop medical products is demonstrate that we're taking a risk-based approach to making design decisions. And we're tracing those design decisions all the way through the process. And we can go on to that a little bit in the following slide. And then at the end of the process, we need to be able to verify that we've met those requirements and that we've developed a product that is not only safe to use, but also functions as it should. Another key difference is that medical products are classified and listed according to the kind of product type and the amount of risk that's involved in them. So an example there on the bottom right, taken from BSI's website, is a handy kind of flow chart that gives you the classification rules in the new in vitro diagnostic regulation. And that new regulation has very different classification categories in it than previous directive. The devices also need to be listed on a central database. So the top right example there is from the FDA's website. So this shows how, in this case, it's a sort of hematology devices are classified under 21 CFR. So they would be listed under, in this example, 21 CFR 864.1860 in the case of the immunohistochemistry reagents and kits, for example. So they're listed in a very formal and centrally accessible kind of way. As an upshot of the new in vitro diagnostic regulation in Europe, the new devices need to be also certified in a different way. So whereas in a lot of non-medical products, it's acceptable for manufacturers to self-certify them as being safe and as meeting the requirements of the EU directives, so the machinery directive, for example, or low voltage directive, something like that. When it comes to in vitro diagnostic regulations, the vast majority now of new devices that will be coming onto the market under that new regulation will need to be certified by a notified body. So it will not be acceptable for product manufacturers to self-certify for a majority of new IVD products. So there's another key difference is the sort of route to market and the route to regulatory compliance. So what does that mean in practice? Well, on this slide, I tried to kind of summarize what various different phases in a in vitro diagnostic or medical device development might look like. So we split it into a number of different phases from left to right. And you can kind of think of that as being a timescale as well, to some extent. Starting off from initial demonstration of feasibility and proof of concept through to prototyping and development and ultimately transfer to manufacture and submission on the right. And at each stage we have design inputs, design activities and design outputs. So in the top left, starting off the process with perhaps an initial concept for a product or an idea, we might start off with the main sort of inputs of that process being a vision of the product. And we might have a high-level product requirement spec or a list of targets, a high-level list of requirements that we think will fulfill the user need. And depending on the architecture of the product, perhaps it's, in the case of an IVD, it might be an instrument with a consumable cartridge, which might have some onboard chemistry or biology element. So there might be some known components already at that stage, and that would all be, the sensitivities of those would all be captured so far as we can in some sort of high-level requirements spec. So the sort of activities that we would undertake in that really early phase of the development, where the technical risk can be quite high, because there's a lot of unproven elements of the product concept, are really aimed at assessing those risks and trying to reduce them, those development risks. So we're spending time putting more effort to develop those requirements and to understand and capture those requirements in more detail, and to develop what the architecture of the system might look like. So where we think the key interfaces are going to be, what kind of interfaces they're going to be, what sort of information needs to be exchanged between various components in the system. At the same time, we would be identifying the key technical risks and trying to, as far as possible, isolate them out so that we can develop those areas and reduce those risks. So we might have a concept for quite a complex end-to-end workflow that's been done in the lab, and the concept might be to try to turn that into a point-of-care desktop system. So we'd be wanting to analyze that workflow and partition it down into sort of sensible chunks, and then develop those and try to de-risk the automation of that process and the miniaturization and the development into this point-of-care type setting.
And I guess just to jump in there, I mean, one thing I'd say is that although we're talking about this in the context, doing that risk-based development in the context of a regulated environment, it also is something that's definitely worth considering, regardless of what type of development you're going to do, because it just ends up being a better, more reliable product at the end of the day, by thinking about the risks very early on at a time where you have infinite flexibility to be able to fix them. Whereas the further you go along down the development process, becomes harder and harder to address those risks.
Yeah, absolutely. And I think a lot of the components of this slide are very common, you know, between medical and non-medical developments. It's just how much effort you put into those various pieces that can be slightly different. So the kind of design outputs that we might have from that initial feasibility and proof of concept phase would be demonstration of those broken down elements and de-risking of those. And also a more detailed list of functional level requirements that we would then feed into the design inputs of the next phase. All the while, actually, I haven't mentioned this so far, but all the while we need to, and in particular with medical devices, I think Dan sort of alluded to this, we have to pay attention to human factors and usability engineering. And in particular, if the product at the end of it is to be used in a point of care setting, and maybe we're aiming for some sort of clear waivable system or very easy to use system, then usability engineering and good human factors needs to be baked in from quite early on. And so in phase one, we might be doing the kinds of studies where we're engaging with key opinion leaders and getting a sense of the sort of market need for the product, and how, while we have that flexibility, we can start to sort of shape the product to meet those needs. And then in phase two, what I've called formative iterations, we're doing a lot more detailed work around human factors, and also the technical development of the instrument and the hardware and the software and the assay and biology might be included in that product. So the kind of human factors and usability work that you might undertake in the phase two would be a lot more kind of user-based. So we might be interviewing potential users and understanding what they do currently, looking at predicate devices and how they work, and you know, how the product that we're envisaging at the end of this process might compete with that, and looking for opportunities there. On the technical side, in phase two, what we're trying to do is develop the alpha prototype, which is effectively a works-like prototype. So it may not look like the final product by the end of phase two. It might look kind of like the phase product. It might have the same size and form factor, but it certainly doesn't have industrial design of the product. But what we aim for by the end of phase two, again, because we're trying to de-risk the development, is that the prototype functions more or less like what we expect the final product to function. So it's really the first opportunity to integrate all of those parts that we developed in phase 1 into an integrated and automated prototype that we can then start to evaluate in a more representative fashion. So the outputs of phase 2, obviously there's a lot of iteration that goes on in that design, build, test kind of cycle. The insights and the output of phase 2 then become design inputs into phase 3, where we're really putting a lot more effort. This is typically quite a large piece of work, the phase 3 product development piece, where we're taking the output of the alpha prototype evaluation and feedback from human factors trials and so on, and going through really detailed development of mechanical design, electrical, and software. By the time we get to phase 3, we would certainly be operating under 13485 compliant quality management system. Quite often that kicks in phase 2, actually. It really does depend on the sort of shape of the project for that particular client and product. But we don't tend to kick in the formal kind of engineering controls until we're a long way down that product development process in phase 3, because we still need to have the flexibility to rapidly modify and change the design as we go.
And I guess one of the differences perhaps between doing that and a conventional product development is there'll be a lot of risks that get identified where it's unclear how frequently those risks may materialize. And with the medical development, if you've identified that risk can be there, it's not necessarily, it depends on how risky it is, but it's not necessarily okay just to say, okay, well, we'll progress anyways and we'll find out about that later. It might be that you want to put some effort into trying to quantify how frequent that risk might actually materialize, whether that's through more detailed back of the envelope calculations or doing some specific tests at the edges of your design space to try and work out when things actually fail. So you get a body of evidence throughout that product development process where you've got a much better understanding of what the eventual design window and reliability space is going to be.
Exactly, and I think there's no coincidence that it's sort of in this picture here, we're seeing a lot of the design outputs of previous phases become design inputs of subsequent phases. And it's the same with the risk analysis. You need to kind of iterate back and forth between identifying the risks, assessing them, mitigating them, and testing that you've mitigated the risk appropriately. So that kind of joined up process is really key to the whole thing. So after phase 3, where we have developed the beta prototype, which is what we would call a works like and looks like prototype, we move on to design, to transfer to manufacture. So by this stage, we should have identified and down selected potential manufacturing partners and really be setting up to transfer the beta design pack to the chosen manufacturing partner or partners. And we quite often get involved in, it really does depend on the shape of project and product, but we quite often get involved in the manufacturing process development as well. And we have an example I can show later on of one of those.
I think the considerations for manufacture are also as equally important as the development itself, because through this development process, you're trying to make something that can be made reliably and consistently at the end. But then a key a key follow-on part of that is who is doing the manufacturer, whether that's TTP or others. Do they actually end up making it reliably and consistently? And that can feed all the way up the supply chain to the specific materials that are being used. And the reason medical grade materials inevitably cost more than normal materials, despite being very similar, is really all about the traceability of ensuring that you're getting the same thing every time at the end, so that once that manufacturer is making that product, you have good confidence that again that device is going to continue to function reliably and consistently in those later stages.
Yep. Anything else to add on this slide before we go on then?
The only thing I think I'd quickly just re-emphasize what you're talking about in terms of human factors and usability, where I think going back to again why regulation and the difference in many products, it's okay to accept that your user is not going to do the right thing, or they're not going to do what you intend, and well, that's not your responsibility as the developer, that they haven't followed the instructions and then it's broken. Well, that's their responsibility. That's not the case for a medical product. At the end of the day, it's the users, if they make users will make use errors, and it is very much the responsibility of the developer to consider those use errors and ensure that for the ones that are going to happen frequently, because we all do things, we all don't read the instructions like we should, that those are not going to result in unacceptable harm, and to test for that really early on. And it's amazing the types of things that you get as I guess the blinkers that you have as a design team when you think about how something should work compared to what you actually see when you put it into the hands of a user as to how they end up actually using it, and the differences that come out there. So it's something that's definitely worth testing early. And again, it's something that at the end of the day, I think goes to making a better product, because you will end up with something that actually people find generally certainly more robust to the use errors, but generally also easier to use.
Yeah, that kind of misuse definition is can be a lot broader when somebody's life depends on the thing being used correctly. Okay, so here's an example of a point of care product that TTP was involved in developing. So this is a desktop system for testing for sexually transmitted infection. It's a PCR machine, gives a result in 30 minutes. The TTP development lasted around 18 months or so. And it went from basically, there was an established proof of concept, but it went all the way from that point. So kind of at the end of the phase one that I showed on the previous slide, all the way through to coming off the manufacturing line within around 18 months. So a fairly rapid, intensive development. It's recently been FDA cleared and clear waived, which means that it kind of by definition needs to be very easy to use and fully automated. So the system, while it is easy to use and fairly simple interface, is very complex inside. You can see there from the top image, there's a lot going on. And there are many sensitive interfaces between the consumable cartridge and the instrument that need to be handled by the design team, and it's doing a high sensitivity test. But the clear waiver requires the instrument to be, as I say, easy to use. The sample needs to be kind of direct to the instrument, so there's no room for processing the sample in any way, or processing it or preparing it. There's no special manipulation it's a sample swab into the cartridge, and that goes into the device. And it's, as I say, it's all sort of fully automated testing inside the machine. The other aspect is the user. It needs to be usable by an untrained user, and give a direct answer as well. So no kind of interpretation required on the answer. It needs to be a clear sort of, a positive or negative, or a clear value, depending on the type of test that it is. So while the development was quite rapid, as you can see, that finished a little while ago now. You can see the amount of time that's passed between then and getting regulatory approval. Gives you a sense of quite how much work is required to get products onto the market once they've been manufactured, and to get them through the regulatory process. So another example, quite different example, this is not a point of care device. This is a much sort of larger system for use in a clinical lab. So this was a product that we worked on. It's for blood grouping and screening. And again, the product itself is fully automated. It gives a very extensive and very multiplexed readout of the blood grouping. And it uses a proprietary microarray, which is shown in the top right-hand corner there, as a key component of that test. Our involvement was to develop that array and the manufacturing processes required to make that. So the middle image there shows an element of that. This is actually quite large, as you can see by the people there. It's a part of a large installation in a purpose-built facility in Switzerland, in a clean room. And it's a complex system that involves automation, delicate substrate handling, high precision motion, deposition of reagents, and online quality control and testing systems, and also integration with various upstream and downstream processes, ultimately leading to the production of that consumable shown in the top right there. So this is a manufacturing system, as I say, that we developed and installed for our client in collaboration with them and other manufacturing partners. And it's being used to produce the consumable for the performance evaluation of the overall product and the submission through CE and FDA for approval. And we're at TTP, we're more recently bringing online in-house manufacturing to enable our clients to get through that kind of performance evaluation more seamlessly from the end of the actual technical development. So as part of that transfer to manufacture piece that I showed on one of the previous slides, sort of phase 4 end of the process, we're starting to bring some of those activities more in-house so that we can provide a sort of a lower barrier and a seamless transition from having a functioning prototype to something that you could actually use for clinical studies and as part of that submission package. And so there's a link there in the bottom left to our website that sort of gives you some more information on that new sort of pilot manufacturing facility that we have. Okay, so 2 examples there of IVD products that we've been involved in the past.
Well, I guess now to switch on to bioprocessing and GMP. So GMP is the regulations that governs the manufacture of drugs and therapies along that line. And I think the very first thing to highlight it is something that governs the therapy manufacturer, not the supplier of equipment. And the reasons for that are because actually through that whole process where we've just been talking about diagnostics and things like that, where you really need that device to function as it should. With a manufacturing line, you actually have the opportunity to put in, to manage the risks in a much more comprehensive way, sort of upstream and downstream of every single bit of equipment. So it might be that you're developing a bioreactor for a monoclonal antibody or something like that. Is if you have a QC test to test whether that's actually producing the right thing at the end of it afterwards, if that thing ceases to work reliably in the way that it's intended, potentially that's just a commercial risk. It doesn't necessarily lead to harm at the end, which is really what this regulation is all centered around. So GMP is really focused on that therapy manufacturer. That being said, when they're using equipment at different bits along the way, that there are many things that overlap and many things in the design and development of that equipment that can actually directly affect that end product. So I mentioned the Davenport disaster, which is what's shown in the newspaper clip on the right, which led to some of the initial, it fed into some of the initial GMP regulation back in 1972, just after the first GMP regulation had been published. And there, again, that was centered around the use of an autoclave to sterilize, I think, drip feed bottles. And there were procedures and processes that were verbally done by the operators of the use of the equipment into how to get that to function reliably, that were not written down, that changed, that wasn't communicated, that resulted in a number of drip feed bottles getting out that were not sterile, that were then delivered to hospitals and did result in deaths and thereon. And that really it highlights the borderline of this this idea that the equipment functioning according to the intended use. So it's the GMP, it's the therapy manufacturer's responsibility to be ensuring that the equipment is doing what it's intended, but the equipment provider can very much help along that process and take on various aspects to help ensure that that equipment is going to continue to function as it should in a way that's safe. And that comes partly down to quality management. It comes partly down to having specifications at the beginning, which are then agreed or between the therapy manufacturer and the equipment manufacturer, and also verification that the equipment will actually meet those specifications and validation. More broadly, there are a few specific risks that are present in a lot of these GMP processing equipment devices. So contamination is very much something that a therapy manufacturer has to manage at all stages of their process. How do they know that the drug product at the end has not been contaminated? Because that, again, can go on to directly cause patient harm, and that contamination can happen at any bit of the process. So equipment that's being developed and used in a GMP environment, contamination is a key consideration that comes down to ensuring both the types of materials that are being used, ensuring, again, traceability in terms of materials. So how do you know that you're getting the same materials in every time? It comes into the manufacturing process for that equipment. I once heard from someone who used to do audits for a regulatory agency that actually, in their view, about 50% of contamination didn't actually come from the materials or parts themselves. It came from the things that were on the around the manufacturing facility and things like that. So they felt that actually a lot of the only way to really get to the bottom of that was to walk around and see the quality procedures in place all the way down the supply chain of where what was going in. So there's a lot that needs to be managed around contamination. Nowadays, a lot of the time you can get suppliers to certify that the materials have been used in a specific way. You can then do the manufacturing in clean room environments and things like that. Related to contamination, but slightly different, is sterility and aseptic closure, depending on what type of process you're doing. But how are you ensuring that, again, if this is a drug or a cell therapy which is not going to be terminally sterilized at the end, how are you ensuring that you do not get bacterial contamination coming in there? And a lot of effort and design effort needs to go into ensuring that that's the case, and then also to test that you can maintain that sterility. Those two risks are I guess, quite related. Slightly differently is how do you know, again, how does the therapy provider know that the equipment is functioning as it should over time, and particularly if it's doing sensing and actually reporting values, and might be very, very simple values, but what's actually being done for calibration and data accuracy? Because again, if your equipment gradually wanders off over a long period of time, that could, depending on its use, end up having a significant effect on the consistency of that end final product. So putting thought into that, and similarly around electronic records and data integrity, if it's something that is taking records of its use and things like that, making sure that those records are traceable and not easily tamperable, so that at the end, when a batch of a drug is made, or when a batch of a cell therapy is made, there's good confidence in everything that has happened along that manufacturing line, and it's traceable if any issues were to arise in the future as to what happened and where were the deviations. So there are standards and regulation that governs specifically electronics and data integrity, and what's needed there. I think just to comment on where it gets a little bit muddier in a way. Two examples that the TTP has worked on that highlight some of the differences. So one, going back a long while, we worked with Amgen to scale out a bioprocess for EPO production, which involved making a manufacturing line of 10,000 roller bottles. And there, many of the things that I've just talked about are exactly the key considerations. So just ensuring, again, the roller bottles maintain sterility, they maintain contamination, and it's a reasonably straightforward thing to do. There are reliability challenges around how you make all 10,000 work at any one time, but actually, because things are being tested, if those reliability challenges come to be at the end, that's a commercial risk. It's not an end risk on safety. It doesn't affect the end quality of the product in a way that can't be detected. On the other hand, we have a spin out at TTP, which is working on doing closed sterile cell sorting for autologous cell therapies. And this is where you're taking cells, it's personalized medicine, you're taking cells from a patient, and then typically you're modifying them and doing various steps to them to turn them into a disease treating cell before you eventually inject them back into the same patient. In that instance, the patient themselves do not, if they do not receive the therapy at the end, particularly a lot of these patients where these therapies are used tend to be end stage cancer patients. If they do not receive the therapy at the end, there's an unfortunate chance and a good chance that actually they will not live to see the next treatment. So the risk profile there, and again, just to highlight, all of this comes back to risk. The risk profile there is potentially very different to what you might see on the left-hand side. And although some of the broad considerations are quite similar, and some of the design considerations and all the things I just mentioned around, does it function according to the intended use? Is it sterile? Are you avoiding contamination? All those considerations are quite similar, but the actual the way the risks can play out in the end can be very different. And so in that instance, actually, that's where we're starting to bring in. So cell therapy equipment is not regulated as a medical device, but there are a number of companies that are straddling that line, and actually some are choosing to develop equipment like they would a medical device, follow the same procedures. Cellular Highways are implementing a 1345 quality system, which is a quality system that governs medical devices to do that. I think the thing that becomes a bit challenging when going down that pathway is it can still be a bit challenging to really identify what is the intended use that you're developing this thing for, because at the end it's going to be used by a therapy provider who will put their own risk mitigations and controls on there. But certainly it can only help with the quality aspects around that and thinking through those risks ahead of time. So in the last few minutes, and I'll just be very, very brief on this because I want to leave some time for questions and things. Wanted to talk about, I mean, often medical device development is viewed as adding a significant burden, particularly a time burden coming on, and it can. And just want to talk about REECE Experience, where at the height of the initial first wave, going back in May last year, TDP and others worked on developing ventilators from scratch. And here we developed a rapidly manufactured ventilator from scratch in around in just under 5 weeks. Whereas part of that, we developed it as a medical device, and we actually developed it with a technical file that I think was certainly hundreds, if not thousands of pages long associated with that, and managed to do that all within the 5-week period. And I think there are some interesting lessons as to what can be taken away there. I mean, the first thing I do want to say is, although it was done in 5 weeks, actually, there was a huge effort. So it was still 10 man years of effort that actually got condensed into that period. But what did make it interesting, and what we were able to do to make it all progress very quickly is to split out a lot of the different considerations that we've talked about today. So we had a design team on one hand progressing that product development in James's original plan, going full steam on there. But in parallel, we had an entirely separate team all thinking in detail around the risks, what's actually going to fail, and trying to identify in quite a lot of detail potential failure modes and then potential mitigation. Similarly, we had a test team thinking about, well, how are we going to verify at the end that this is actually working as it should? A human factors team that was going on the side doing the user interface along the way. So by splitting all of this out, we're able to move a lot more quickly while still getting the benefit of the quality and the risk management that came through. I think I mentioned it was about 10 man years of effort at the beginning. I think around 25% of that effort, well, 50% was on the core product development, around 25% was on the quality, the risk management aspects and things like that. And then the remaining 25% was on a range of different aspects like the transfer to manufacture and aspects like that. So it's possible to arrange a program in a way where you can have different teams almost checking each other's homework as a way you go and move quickly. But with that in mind, we'll just try and wrap up with a few just quick top tips and then open it up for questions.
Yeah, okay, so I'll kick this one off. I think tip number one is try to consider your regulatory strategy early. So obviously you don't need to have a fully baked regulatory submission plan from the word go, but at least have good idea of what your intended use is. That allows you then to categorize your device and start to think about what the key risks are likely to be, and you can build a risk mitigation plan around that. As Dan said before, the kind of regulatory process, or the development process that we undergo in a regulatory environment, it's all kind of generally good practice and sensible activities to be doing in any case. So actually in those early stages of the development, there's not too much of an additional overhead to make a start on some of these items. So it's a good idea to sort of bake them in early on if you can. I think also another thing, which is obviously good practice from a product development point of view, is to try to get feedback as early as possible from the market, from key opinion leaders, and really try to, qualify and justify the value proposition, and be agile. Don't be too wedded to that original vision. Make sure that you're open-minded and happy to change direction early on if necessary.
Another one I've seen is, and just advise, is really be guided by the risks. Don't focus too much on the process. I've seen worked on projects where the process has ended up being the driving aspect behind everything we do, which leads to a lot of work and overhead and things. At the end of the day, it is about risk, and actually I think that can lead you down some quite creative design decisions. We worked on a life-saving drug delivery device, where if we'd just done things in the default way, it would have led us to a very expensive software development of trying to think about how we can make some challenging software extremely reliable and validate every single function, and that would have incurred a lot of cost. But actually, by stripping it back and thinking about what were the risks if the software goes wrong, can we accept certain risks? What risks of the software going wrong can we accept, and which can we not accept? We were able to actually even think then about our electronics architecture and split the software across different bits of the electronic software architecture, put in redundancy in the electronics, and really shortcut and simplify that whole development process. And in very practical terms, move down a classification in terms of the software development, but still feel equally comfortable and confident with the risks being managed at the end of the day, which is the goal.
Yeah, I think another one I would say is that another point I would make is that regulatory bodies want to get life-saving therapies, diagnostics, technologies onto the market. So they're on your side, and they're more than willing to interact with you fairly early on in the process. And there's lots of sort of education and learning resources available around the submission process. So I've included a couple of URLs there for the FDA and also the European organizations. But yeah, there's a lot of resources available, lots of other sources of information, and yeah, like I said, they're on your side. Talk to them early.
With that, we should probably got a few minutes. If there are any questions, I'd love to answer some questions.
I'll just quickly interrupt there. So thank you very much, Dan and James, for that very comprehensive overview and some really interesting and insightful case studies there. Quite a broad range of technologies you covered. So that's really interesting and hopefully something relevant to everyone on the call. I can't see any questions come through as yet. There is still time. So don't be shy. That's your perfect opportunity to talk directly with the experts on the line. One thing I'd be interested to know is, obviously in our field where the pace of innovation is very fast, and perhaps regulation and guidelines are changing, what are your tips for developing products in a space like that?
I think, so the autologous cell therapy space is very much a good example of that, where it's one where the therapies and the developments that are happening with therapies are really happening as faster than the regulation can keep up. And it does make it challenging from actually from a designer perspective, and that there are questions and things you're wondering about how certain things are going to be received, whether you have to do something or not, and there's not a clear document you can go to and a clear answer to say this is what you need to do. The thing I would suggest again, is again, be guided by the risks. So it comes down to making an assessment of what those risks are, and are they trying to think through, just in the way you would do any type of regulated development, are they likely to be, is the risk benefit likely to be acceptable? And if you can do that, that's the sort of judgment that the regulator is making at the end of it. So it's not to say that you will have the same opinion at the end, but it certainly puts you in a good place. The second, I think, is if you can try and get guidance from the regulator early, and they are quite, they can sometimes be quite happy to talk about different innovations and what their views of how things will play out on that, and that guidance can be invaluable. It doesn't always happen. I was on a project where we asked for guidance, and we were told instead that we had to wait for 6 months to a year, and then they would publish some formal guidance, which didn't really help in the context of a 2-year-long development. But it's definitely a good place to start. James, do you have anything to add on that?
No, I think, again, it's that last point, isn't it? Talk to the regulators if you can.
And another thought. So when we talk about sort of hardware projects and even software, you often hear the term agile used quite frequently, as in sort of take an agile approach. How does that fit with a tightly regulated, controlled environment?
So I think, maybe if I can get back to this slide here. Yeah. So the agile approach definitely fits more easily into the early stages of the development process, where you have a lot more design freedom, and you're doing the kind of formative iterations and sort of feasibility demonstration. So that fits well, I think. Where it doesn't fit so well is when you're under more heavy engineering control and your designs are frozen. Kind of by definition, it's much harder to make on-the-fly changes and be agile at that point. So there is definitely a tipping point. And I think that's typically between the sort of phase 2, phase 3 point in this kind of structure, where agile works well and becomes sort of less easily transferred into the process.
And I guess one of the things with that is often with a consumer product with an agile development or something, particularly if it's a software development, you know, the part of the reason why you're doing that is you're doing the agile development so you can test it more rapidly with your end customers and iterate. And as we were discussing before the webinar, change Zoom on a daily basis and get it to where it needs to be. Perhaps one of the differences, if it's a true medical device, is you won't necessarily be able to easily, rapidly, and iterate with your end user base. Although you can still get a lot of that insight in different ways through formative studies and testing and things like that, which I still think taking that rapid iteration approach can really help bring good insight into the design and development process early on. It's just that you might not be doing it with your end customer or the patients at the end who might be who could potentially be harmed in the process if it were actually hadn't gone through all the testing and things that needs to happen.
Yeah, and I've throughout the presentation, a very consistent theme has been that emphasis on the patient, sort of the importance of why we are so particular and careful in these environments, because ultimately it's been used sort of for that purpose on a patient. So yeah, a really important point to highlight there. We are up to time now, so I think I'll probably close the session there, but I'm sure you'd be very happy if questions come through, to those attendees on the line still. If you do think of any questions or you would like to get in touch directly with either Dan or James, thank you, Sophie, for your comment. Thank you. I'm glad you found that informative. I'd be very happy to help facilitate that if you don't already have the connection there. So please do get in contact. There'll be the post event follow-up that goes out, so you can use that to reach out if you'd like to, and that will also include a link to the session recording. So with that, thank you once again to Dan and James, and thank you to all the attendees for joining. And I'll close the session there and wish you a good evening. Thanks very much, everyone.
Thanks very much.
Cheers. Thanks.